Institute, our neighbor down the street. She earned her PhD in physical and biological oceanography at Lagos in Toulouse, France, and came to MBARI as a postdoc and then a research associate in the Francisco Chavez's biological oceanography lab before becoming a research specialist working across different labs at MBARI. She's also a Marie Curie Fellow, spending time at the Mediterranean Institute of Oceanography in Marseille. And her research combines a number of different techniques: in situ data, satellite products, numerical models to study ecosystem projects, and physical biological coupling in coastal and open oceans. So we're looking forward to hearing more about that today. And let's give a big welcome to Monique Messi.
Thank you, Tom, for the introduction and the invitation, and thank you all of you for being here. It's actually great. I'm just down the street, and I don't come here very often, so I'm very grateful for the opportunity to present my work. And so today I will be telling you about what I call coupling and decoupling between upwelling and biological response in Eastern Boundary Upwelling Systems in general, but mostly in California, right here. And so a lot of the research I do kind of looks at broadly the question of what drives biology in upwelling systems and more generally. And I like that picture of Monterey Bay because it shows kind of a biological hotspot with whales, sea lions, seabirds, and even humans. And so what I'm trying to do is to understand where and when we have those intense biological activities, and mostly why. And for that, I'm mostly looking at physical drivers. And the reason we care about it is that we know, I think, more about physics than we do about biology in the ocean, and we still don't fully understand the physical processes that drive some of that biology. So a lot of it is, especially in the California current system, bottom-up driven, which means that the reason for the productivity we have here is directly linked to the plankton. And this is an image from satellite. It's ocean color that shows chlorophyll concentration of the coast here. And so you'll recognize Moss Landing right here and the San Francisco Bay. So this is the whole California current system that's extremely productive with a lot of plankton. And of course, the reason for that is upwelling. This is a little schematic that just shows you how upwelling drives the deep, nutrient-rich waters into the surface, where it stimulates phytoplankton, zooplankton, and then that propagates all the way to the food chain with fish and whales and top predators. So we've known for a really long time that upwelling is the reason for the high productivity we have here, but how that forcing is actuated, like what is the true link between the two, is a little bit harder to look at. And so I'm talking about upwelling a lot, but really what the process is that nutrient supply, which is the product of upwelling as the amount of water that's being brought up by the surface, which you can estimate from the winds, to nitrate concentration at depths. And so I use 60 meters. So the product of the two is basically the amount of nitrate that is being brought up to the surface and that is available for primary production. So I'm not going to talk about fish and whales. This is a different view of the same system. We have also upwelling coming up near the coast, and then those waters moving offshore with first phytoplankton, zooplankton, and some carbon exports that brings that productivity down through the midwater and benthic communities. And so I'm looking at that system in terms of phytoplankton, zooplankton, carbon exports, and midwater and benthic animals. So those are the components of biology I'll be focusing on today. And so the question I'm asking is whether we can observe and identify the response of the systems to that upwelling process in the California current system. And it's actually not that simple because this is a first example, and I will explain later how I get those time series. But basically, you're looking at temporal variability of surface plankton here, midwater animals here, and benthic animals here at 4,000 meter depths. So throughout the water column. And we're trying to relate that variability to upwelling as it is represented by those time series. Here, this is calculated from in situ winds at a mooring just out of Monterey Bay. And we're looking at daily measures of that upwelling process, the influx of water into the surface layers. And for the benthic time series, which is located in this location at Station M further south, I'm comparing it to the Point Conception upwelling. So in other words, there's no obvious links between the two. And so if we want to look at the link between upwelling and biology, the first thing that you might notice is that the variability is very different. So maybe something we can do is to actually smooth the upwelling signal. Instead of looking at a daily time series, we can look at a monthly time series. And sometimes it works fine. Like plankton is an example where we do get some significant correlations between the two. So you can say, sure, upwelling drives phytoplankton. But if you look at midwater and benthic animals, there's just no correlation between those time series. So the answer is not that easy. There's really a temporal decoupling between physics and biology that's kind of hard to explain from those simple patterns. There's also a spatial decoupling happening. I really like that figure from a paper by John Ryan that shows sea surface temperature on the left. And so basically, wherever you have cold waters, you have a lot of upwelling, and ocean color sea surface chlorophyll on the right, which is an indicator of phytoplankton. And so at first glance, you would say that they're really linked. Like when you have cold water filaments going offshore, here you have higher chlorophyll. You have higher chlorophyll near shore where there's a lot of cold waters. So at first glance, maybe upwelling seems to be correlated to the biological response. But if you look a little bit more closely, that's not actually true, because the coldest waters here are kind of linked to low chlorophyll, and the highest chlorophyll here are for intermediate temperatures. So there is a spatial decoupling happening. And so basically the goal of this talk today is to explain those both temporal and spatial decoupling that we see between upwelling and biology, and kind of look at whether upwelling does drive biology spatiotemporal variability, and if yes, how? What are the mechanisms that link to those two? And so I will be talking about a couple of processes. The first one is persistence, and for this I will be talking about the time series I presented at the beginning, so surface, midwater, and benthic animals. And the idea is that when you have a forcing such as upwelling, biology may respond right away, but maybe that response actually lasts for a certain amount of time, so that when you stop upwelling, you still have a response. And so that's what this little graph represents, where you have a response as a function of time that maybe is maximum when the forcing happens and then decreases over time, but does persist for a certain amount of time. The second process that can explain some of those decoupling is lags. And so in this case, it's what happens when you don't have a biological response that's immediate, but it's actually shifted in time, so that if you, same thing, you have the forcing here, and that response is actually a little bit later. And that's particularly important in a place like around here where the currents are very strong, because if you have a certain amount of time between your upwelling at the coast and then your biological response, you're going to be moved offshore and with the currents, and so that's going to happen in a different location. And so for that, I will be looking at zooplankton and carbon export. And finally, what I'll do at the end is kind of talk about how we can use that knowledge to extrapolate what we learned from the data in the California current system to other systems across the world. And that's very much a work in progress, so that, that's a disclaimer that it won't be the truth, but just kind of an intellectual exercise to look at the response of biology across systems. Okay, so I'm going to start with the persistence, and so go back to those time series I was presenting at the beginning, and now explain a little bit more how they were collected. So as I said before, surface and midwater were collected here in Monterey Bay. The benthic was collected here at Station M. And I'm also looking at the time series from those two moorings here and there to match upwelling to the biological response. So the time series were started a long time ago, 1989 for the surface plankton and the benthic time series, and 1997 for the consistent data for midwater, which is what I use here. Bentic was actually started at Scripps, and then the PI on this, Ken Smith, came to MBARI in 2007 when MBARI took it over. So for the surface, we looked at water samples collected from a CTD rosette. So this is the Rachel Carson that maybe you've been seeing in the bay. They go out every month looking at 3 stations. I'm just looking at data from 1 station here. And so we collect water at different depths. Here I'm only looking at the surface samples that were collected around 2 meters. And so that water is brought back to the lab and is being looked at under a microscope. And all of the plankton is identified, counted, sized. And from the size, you can estimate their volume. And you're looking at relationship between carbon per volume, you can estimate the biomass of the plankton. So that we have any estimates of plankton biomass as a function of time for different groups, different taxa of phytoplankton and some zooplankton. For midwater time series, we've been using ROVs. And so those are transect where the ROV stays at 1 depth and kind of moves at a constant speed for 10 minutes at the same depth. And we have cameras that image everything, and I'll show an example of that a little bit later. And so you can estimate the volume of water that's actually imaged by the ROV based on camera specifics and light penetration. And so if you look at all the animals within those transects, you can divide that by the volume and get the density of animal per meter cube. We have transects every 100 meters from 200 to 1,000 meters. There's a couple of shallower ones that I'm not using. And here I'm averaging or integrating all of that data together, so there's no information on depth variability anymore. It's just a measure of all of the animals seen in that depth zone. And finally, for the Bentic time series, it started using towed camera sleds and then moved to ROV video with MBARI in 2007. And so it's basically the same principle as for midwater. The only difference is that instead of imaging the open water, you image the bottom, so the animal density you get is as a function of meter square of the seafloor. There's a lot of diversity in the ROV video, but because we wanted to use long-term time series, we kept only the animals that were seen reliably in those camera sled images. And the other difference also between the Bentic and the other two is that those were monthly, whereas this one was only about twice a year, because it takes a long time for the ship to go all the way to Station M and do those ROV transects, so it's not something we could possibly do every month. And so this gives you an idea of the data that we're looking at for a midwater transect. And there's a lot of marine snow in that image. And so the ROV is just horizontal, which is the reason why you're not seeing the vertical movement of that, just because the ROV speed is faster on the horizontal, but it is also happening. And there's a lot of animals that it's kind of hard to see, and I would not be able to tell you what they are because I'm definitely not an expert. But sometimes, people in the video lab look at them, and like this big one, for instance, was a larvacean with its house that they can identify and then annotate. This is something similar for the Bentic time series. That was one example where there was actually a lot of animals. Normally it's a lot more bare. Those 2 red dots that you see can be used for sizing. Those are laser pointers, but there was no sizing done for this specific project. And so, so those are the data that we're getting back from the ROV. And then people in the video lab at MBARI look at those videos, slow them down, look at frame by frame, and then identify the animals and annotate them. And all of that is done in a system that's called VARS. It's the Video Annotation and Reference System. And so we have a database of all of those annotations. And that's kind of the animals that the different time series represent. So for the surface, we have a mix of very small plankton, like Synechococcus, that's a picoplankton, and some larger ones, like dinoflagellates and diatoms. And for the midwater, it's a lot of gelatinous animals. That's the big difference between what you can see with an ROV and what you would see if you were using a netto, for instance, because we get all the gelatinous animals very well. There's also some squids, some crustaceans, some fish. And for the benthos, there's only 10 species that were reliably annotated throughout the time series, and they're mostly echinoderms, which means sea cucumbers. So I'm not expecting you to actually look at that whole graph in details. That's just to give you an idea of all of the data that those time series represent. And so the way to look at those graphs is that the x-axis is time, the y-axis, each line is one species, and the color of the letters indicate which taxonomy group they belong to. You have the caption here. And each of the colors in the middle of the graphs is an anomaly of the data, so surface biomass or animal density. And we put a square root transform to it because they were very variable. But besides that transformation and removing the mean, everything else is in there. There's a few patterns that you can see already by just looking at those. One is that the surface values are a lot more variable than the others. Some of it is because the temporal resolution of the benthos, as I said, is a lot lower, but even then, like, after 2010, you just have positive values here, whereas surface tends to go up and down a lot more. The other thing to note is that there's a lot of synchrony across the one time series, like that's most visible in the benthos, where most of the animals have red colors, meaning higher than normal after 2010, and low colors in around 2007, meaning lower than normal. And even midwater that has, about 90 species, you can kind of see a lot of animals having negative anomalies, so lower than normal abundance before 2000, and then higher than normal after 2010 or so also. So that's too much data to look at, and so we wanted to synthesize that. And so what we did is that we took advantage of that synchrony across taxa within one time series here to extract one time series that's representative of the whole community. And we did that using principal component analysis. And so what we get is just one time series that represents the most variable, the most variance of the original time series. And so in this case, you can explain about half of the variance of the original data set using that one time series. And you can see that the values correspond to what you see in the original data with a lot of positive, higher than normal values after 2010, very negative ones around 2007, which is what I was pointing out beforehand. So we did that exercise for the 3 communities, and so that's how we ended up with those 3 time series. And the percentage tell you the percent of variance explained, which is highest for the benthic and lowest for the midwater. And some of it is just because there's not a lot of animals in the benthic time series, and there's a lot of animals in the midwater time series. The interesting thing about it is that there's no inter-community synchrony. All of those time series are very different. Like the scale of variability is different. And even for the midwater and benthic that are similarly kind of like slow moving, where you can really see long times of like low values in both time series, but they don't actually match that well. Like for instance, in the benthic time series, those were mostly positive, whereas they were negative here for midwater. So there's not really any obvious link between those time series. And when we started this project, actually, we were looking for a link between the time series, and we thought they would be, forced by something simple like El Nino or the climate indices that we have that represent variability around here, but there's really nothing there. And that's where we went back to upwelling as a potential driver and tried to look at ways where those upwelling time series maybe actually drive the biology, but in a more complicated way than just coincident forcing. And there was kind of like that aha moment when we came across that paper by Di Lorenzo and Oman in 2013, where they talk about the concept of integration, or that's what I was calling persistence earlier. And the idea is, the best analogy is maybe music, where if you have somebody playing music with a harp, for instance, they're going to pinch the chord, that's going to make a sound, and the sound is going to last for a certain amount of time. It's going to be strongest just after you pinch the chord, and then you will hear it for a long time, but that's going to decrease over time, which is really what that little curve represents. And the damping time scale is kind of that characteristic timescale of how long you can hear the note. And the reason why it really changes the signal is that if you don't play just one note but several, then you start to have all of those notes getting superimposed and creating that melody so that what you hear at a given time is not just the result of what just happened before, but what happened in a certain amount of time prior to the time when you're listening, because you have all of those notes that you're still hearing. And you can actually represent that mathematically. It's called cumulative integrations. And I'm not going to go through the math, but just to show you what the result is, here you have in gray a time series of upwelling over 2 years, and in colors you have 2 different integrations using 2 different time scales. The green is just 1 week, and the blue is just 1 month. So again, that's kind of like that characteristic time scale, but it doesn't mean that your signal is going to only persist, for instance, for a week here. It still persists longer than that, but it's just basically that e-folding time on that exponential decrease. And the main thing about this is that the longer integration timescale you have, the more you smooth out the signal, so that if you have 1 week integration, you still see a lot of the original signal in it. If you go to 1 month, you still start to pick out more of that very low frequency variability. But you still see all of the impact of the individual events. For instance, if you look at that one upwelling event at the beginning of April, you have a very strong peak in the green at the same time. I'm looking at April 2022 right now. And you still see a peak for the 1 month integration line, even if it's a lot lower and a lot less visible. So basically what we did is that we applied that mathematical equation to calculate upwelling integrated with a bunch of different timescales, and we looked at the results against the time series of biology that I was showing you just before. And so if I go back to my 3 time series, and here is the example of Monterey Bay upwelling that I matched with surface and midwater, this is what we get in terms of the best time series, the ones that explain the most the biology represented in those colored time series. So for surface, we found that a 16-day integration was the best. For midwater, that was a little bit more complicated because there's so many animals that there was not clear defined peak. But here I use 4.7 years as kind of a good representation of something that works pretty well. And for the bent oak, there was a peak at 4.5 years also. Something that's interesting to note is that I'm using the same integration for midwater and bentos, but the black lines are different. And that's because the midwater black line is based on the Monterey Bay upwelling time series, but the bentos black line is based on the Pont Conception upwelling, which is a lot further south than here. And so there's actually significant differences even within the same region. You can get some pretty different variability if you're in a different location. And so those integrations actually explain quite a bit of the variance, because we can go, surface is 17%, it's not a lot, it's something that's very variable, it's a little bit harder to explain, but we can explain 34% of the variance for midwater and 58% of the variance for the bentos. So if you consider that those principal components actually already explain a smallish percentage of the variance, it's not that high overall, but it's the one forcing that we found that explains the most variance of the data. And again, we looked at other in situ variables, whether temperature, oxygen, or salinity, large-scale climate indices. We spent a lot of time before we found that link to try to explain those time series. And so what this actually tells us is that, yes, coastal upwelling does drive ecosystem temporal variability, and that's seen at the surface, but also all the way to the seafloor at 4,000 meters. So that those animals respond to upwelling, but there is that damping timescale that transforms the signal. So the next question is, what explains those 16-day, 4.5 years? I will skip the 16-day right now. I'll actually show that a little bit later. But for the midwater and bentos time series, it's actually linked to animal lifespans. And that was what was originally proposed in that 2013 paper, and that's something that we believe is happening here as well. And if you think about it, that kind of makes sense, because if you have an increase in the number of animals that you see because of upwelling, that's going to last until those animals die. So if you look at individual time series, this one is a small medusae called Aegina. And so here the colors represent that temporal variability of Aegina, and the gray line is the best integrated upwelling that explains it, and it works with 9 months integration damping timescale. Whereas for something like a fish, which is actually I learned during this project is the most common fish on Earth. In this case, something like 4 years is what represents that variability the best. And so that makes sense with what we know of those animals. Those are extremely hard to validate because we just don't know anything about how long animals live in the ocean. And so that kind of gives some ideas, so that can be useful for biological purposes as well. And just to summarize with this first part, the persistence is something that transformed the original forcing. And so you can represent it by mathematically using those integration. And how that transformation happens, meaning what the damping timescale is linked to animal lifespans. So I'm going to switch gear a little bit now and talk about lags. And so for that, I'm looking at very different data, which is data on zooplankton and carbon export. And so the reason for that lag is that you have some dynamics in the plankton that causes some of the plankton members to take more time to respond to upwelling than others. Because when you have an input of nutrients at time 0, this is the output of a little plankton model that predicts that phytoplankton grows very fast and then is being grazed by zooplankton that takes more time to respond, so that actually the zooplankton maximum response to upwelling is about 3 weeks after upwelling happens. And in this case, zooplankton was parameterized based on krill, and we were using that little plankton model to also look at carbon export as a function of zooplankton. The idea is that zooplankton fecal pellets is a very big contribution to carbon export in the California current system, and so you can consider that that carbon production from zooplankton is linked to export. And one thing to note, I was talking about the 16-day earlier, is that if I overlay my damping timescale schematic from before, and if you look at that blue line versus the black line of the phytoplankton, it's actually not that different. And that's the reason why the integration was working pretty well for phytoplankton, because realistically the function as a like that line, like the response as a function of time, is pretty similar whether you predict it using a model like this or if you're just doing a statistical integration. But this part is not about phytoplankton. It's really about the ones that do show lag, meaning zooplankton and export. So why that matters and why we really need to take that into account is because of advection, which I mentioned before, so that when you have upwelling at the coast and you consider the water masses are moving around, and now you're looking at something that happens 3 weeks later, you can be a lot further offshore. And so in order to kind of understand that relationship, we used a data set of sediment traps that is from the CCE-LTER program out of Scripps. And so those are sediment traps that are deployed at the base of the euphotic zone and drift for a few days and just measure the amount of carbon that sinks. And then I used surface currents that you can get from satellite to backtrack where the water masses were coming from at that location of the sediment traps. And so that's what all of those gray lines show. They connect each sediment trap to its coastal origin. And that can be, like a few days before, or for some of the sediment traps, that's actually weeks before. And so you really need to, in order to understand the response of biology for those type of variables, you need to look at it in a Lagrangian framework, which means you need to look at it along current trajectories to be able to understand the link with the original forcing. And some people have done that. This is a pretty recent paper also out of Scripps where they were looking at the response of diatoms and copepods as a function of time following upwelling. So it's actually good because they find something similar to that NPZ model I was showing before, with diatoms responding first, and then copepods responding, a little bit later, like around 3 weeks again after upwelling. And so for the export data set, I did something similar where using those trajectories, you can get water age, which is the time following upwelling, like how old is that water mass since it's been upwelled near the coast. And here the color represents nitrate supply at the coast. And so basically, export is a function of both how old, like how much time has elapsed since upwelling, and also how much nitrate was input at the beginning. And if you look at if you take that biological response function and you scale it with that nitrate supply, you can get something like this on the right, where each one of those curves is the response of the model for a given nitrate supply. And if you combine the nitrate supply at the origin of the sediment traps with that model, and that's the y-axis here, and you compare it to the in situ measured export, that's the x-axis here, we actually can explain almost 70% of the variance, which is really big for something that's in situ biology compared to something that's purely satellite derived. Those 70% do exclude 3 points, those 3 that are circled in black, and those were shallow traps because the surface currents that I'm using are representative of the top 30 meters. So if the sediment trap is within that region, that actually doesn't really represent very well the water mass advection above the trap. So I believe that's the reason why those 2 points don't work. So now we have an expression of how zooplankton and export respond as a function of time and nitrate supply at the coast. And so you can do the other one in reverse. Instead of backtracking, now you can go forward, which means that you can start from the coast and the plankton model to see what happens afterwards. And that's what I call the gross advection model. And so basically the idea is that you have that simple plankton model, and here we're really just looking at that black line that is forced by nitrate supply at the coast, which is estimated from satellite, and then we follow the currents. And so just to give you a little visual of what that looks like, I'm looking at evolution over time following upwelling at the coast, and that's something that runs for about 60 days, I believe. And the colors in the plot basically show you the concentration of plankton predicted by the model, where when you have high values, it's because the coastal upwelling was highest, the nitrate supply input at the coast. And you can see how those currents kind of like take those water masses and move them in totally different locations. And so what you do then is that, so you have maps such as this, which is, like 1 day, like initialized on 1 day. You run this every day, you combine everything, and you can create gridded maps. And so here I'm representing zooplankton for 1 month in May 2008, and so that combines a bunch of these different Lagrangian runs like this. And so you might be wondering how well something like that works, because we're used to more complex coupled physical biological models. And so we did a little exercise where we were comparing the result from this model to a Rum's-Nemiro model, and we were forcing the gross advection model with the same fields. So it's just a proof of concept, because the only processes that are being included in the gross advection model are coastal upwelling, plankton dynamics, and advection, whereas Rum's-Nemiro-CSC is a lot more complicated and has a lot more processes in it. But we actually represent both the spatial patterns and the temporal variability of krill hotspots pretty well. This one is not as good, but that's simply because it's all the way in the north of the domain, and that's going to be influenced by a lot of waters that come from outside of that domain, which obviously we can't get since I was only using this domain to force it. So that works pretty well when compared to a model. That also works when compared to data. And again, that's a model that only includes coastal upwelling, plankton dynamics, and advection. And so I can, compare that to time series of in situ krill surveys collected by NOAA in May and June of the coast of California, and show that we explain quite a bit of the variance of those time series. It's not significant for the southern hotspot, which is the one right here. There's just not a lot of data. So the data here are just the black lines. And so as you can see, there's just not a lot of them. But for the central hotspot, it actually works pretty well. And 40% of variance explained is not huge, but for biology, I actually consider it to be pretty good overall. And same thing with export. If we use the same model and we look at export, the difference between this plot and the previous one is that the previous one was only using those trajectories, so only backtracking a single, like getting a single point for each sediment trap. Now I'm comparing the same in situ export to the grid points there. And so we're looking at something that's 1/8 of a degree resolution, so that also includes more data. So the correlation is not as good, but it's still not bad, and it compares fairly favorably to another ocean color-derived product of export from satellite that's basically just saying export is proportional to primary production. And what's interesting is that the best value is actually when you combine the two, where you can get as high as 2/3 of the variance explained. And the reason for that is that the ocean color one is mostly based on the phytoplankton contribution, whereas the gross advection one is mostly the zooplankton contribution. So if you look at both of them together, that actually increases the how well you can predict export from satellite. And that has implications because they're very different patterns. This is an example for the same months of ocean color-derived carbon export, and the one from the gross advection model. So the patterns are different, and the variability is much higher in the gross advection model. So that it does matter to include that when we look at estimates of carbon export from space. I'm going to skip over this because I'm running out of time a little bit, but I can talk about the link with deeper export of carbon to the seafloor as well. That can be useful for that model. And so just to finish with that second part, this process, the main effect it has is to displace the original forcing, meaning you have a latitudinal variability like this of coastal nitrate supply, and the maximum production that you might have, in this case I'm looking at export, is going to be moved further offshore, and it can be offset in latitude as well. And so both that the temporal lag depends on the plankton dynamics, and the horizontal lag depends on oceanic currents and where they're going and how fast they're going. One thing I should have mentioned is that when you look at this, it has to be plankton. Like, you couldn't look at that framework for midwater and benthic time series because the animals can swim around, and so they're not as impacted by currents. All right, and then in the last few minutes, I'm just going to talk a little bit about implications across Eastern Boundary Upwelling Systems. And so this map just shows you where California fits in relation to the other major upwelling systems, which are Peru, Northwest Africa, and Benguela. So California is actually the least productive of the four, but all of those are kind of driven by similar processes of coastal upwelling. And so what we've been doing is to try to use that knowledge gained from what I've been talking about so far to kind of extrapolate the biological variability across the California current system, but also the other ones. And so it's a pretty simple process where you take that response function and you just apply it to satellite-derived upwelling at the coast in all of the systems. And similarly, you can run that gross advection model using satellite-derived variables in other systems. One difference with what I was doing in California is that in California, I was using climatological in situ nitrate. You can't do that in other systems, especially Peru, that has a lot of internal variability. So I've been using outputs from a European model that gives me nitrate. But the rest of it is satellite-derived. And so just briefly, I'm happy to talk more about this. It's just to give you an idea of what that might look like. This is the graphs for all of the upwelling systems as a function of time on the x-axis, latitude on the y-axis, and the colors represent, in this case, coastal upwelling. So that gives you an idea of the latitudinal and temporal variability of that process. So you can see things like, Central California has a lot of upwelling. There's a lot of seasonal variability that you can see. And so the strength of it depends on the system and all of that. You can spend a lot of time just looking at those. It's just to give you an idea of how variable it is. And then I ran the integration to look at potential response for marine animals. Those are yearly averages. We don't really know what the mean state is, so that's just anomalies, which is the reason why those are between positive and negative. I removed the mean at each latitude so that I have no information about the latitudinal variability of those animals. It just gives you the temporal variability. And so if you remember, we were around here, and so we had that kind of positive trend, which you see again here. But that's not what we see in the other system, except perhaps Benguela. Something like Peru might have been like low, then high, then low. Something like the Northwest Africa might have been low, then high, and like a lot more variable, so that what we see in one location is not representative of what happens in the other systems. And of course, the main issue with that is that we have no way to know if it's real or not. We have no other models that measure, like really represent the response of marine animals in other systems or even across California. So it's purely speculative and a way to look at, what would be the implications of the upwelling system changes across the upwelling systems. And if you do the same thing for krill, this is a lot more similar to the original upwelling time series just because krill responds a lot faster. So when you're looking at those yearly averages, it kind of smooths variability out. You do have a displacement. For instance, Peru upwelling was strongest at the northern end of the domain, and that's getting shifted further south by the currents. And there's also some clear interannual variability that's happening here, but overall it's higher frequency than what you see with the marine animals. And same thing, there's not a lot of information or time series on zooplankton in other systems, so it's really challenging to validate those. For this, it would be interesting to look at other coupled physical biological models in other systems, which is not something I've looked at yet. All right, so I'm going to stop here with just a few conclusions. So what I hope I've been able to show you is that the upwelling and biological response can be decoupled in time because of those two processes, the persistence and the lag. So that depends on which variable you're looking at, whether you're a plankton or a marine animal, but both of them should be taken into account when trying to relate a biological response to upwelling. That time decoupling can lead to space decoupling for the plankton because of the advection by currents. And both of those processes can change the nature of the biological response to physical forcing, which means that if you start from a very variable time series, maybe you're going to end up with something a lot lower frequency. You might have a forcing in one location and actually see the response somewhere else. So they need to be considered to be able to predict the biological response to upwelling. And finally, I want to just acknowledge a lot of people who contributed to the different studies that I was just showing you, and funding coming from so MBARI, the David and Lucille Packard Foundation, and NASA. And I'm very happy to take any questions.
Thank you, Monique. We have time for questions, and I will, for questions that are in the room, I'll pass around the microphone, and this is for the people on Zoom. It's not going to amplify your voice, and then we'll see if we have any Zoom questions as well. So any questions?
Hi, I had a question regarding process 1, specifically the midwater data set. Did that data account for any species that might vertically migrate, like at night?
That's a very good question. All of those were transect taken during the day, and so we did not look at the diurnal vertical migration. But since we were consistently at the same time of day, that should account for that. The fact that we are restricted between 200 and 1,000 meters, there are some species that their range was a little bit, like might go a little bit above 200 meters, for instance, and so we might not capture that perfectly. But yeah, that was kind of the best we could do with that data, I guess.
Hi, thank you for your presentation. That was super interesting. You mentioned that you use sediment traps to record the carbon, the sinking carbon. Have you looked at, or your team looked at sediment cores, long-term impacts of those measurements?
No, I have not. There is, so the data that I didn't have time to show was a sediment trap much further down in the water column at Station M, where we had the time series of benthic animals. And so we actually have sediment traps there that are 600 meters above the bottom and another one 50 meters above the bottom. And so there's been more work trying to relate the carbon flux in those traps to the response at the bottom, and that includes not only animals, but there's also measures of respiration in the sediments, where there was a rover that was going around and lowering basically a device into the sediment and measuring the oxygen consumption within it. So it's not something that I've participated in, but I can, there's papers on that from Ken Smith's lab.
Thank you. I was curious about this lag and advection model. That is an impressive amount of variation explained. And I was curious, you said that it could not be applied to higher trophic levels because of their mobility, right? So, but presumably also they might chase the food,
Yeah.
the plankton, right?
Yeah.
So did you do any exploration trying to apply the same logic to, say, fish or higher levels of the trophic cascade?
I'd love to. I think what would be interesting is that conceptually you don't have to use the NPZ model I had. You can use any type of function, including statistical. So the problem is I don't think we have really the data yet to look at that, because you would need something a lot more distributed than what we have. And I'm not aware of a data set that could be used to actually force that, but conceptually, if we had the data, yes. And if you have ideas or suggestions about data sets I could look at, I'm definitely interested.
No, I actually was thinking about the data set you already have, because you had that, long time series.
Yeah. Yeah, and it's just 1 point. And I think the other problem is that that model works pretty well offshore, not as well inshore, so we could look at it for Station M data, but the midwater animals in Monterey Bay, the satellite currents I'm using are just not good enough near shore. So now with SWOT, maybe we'll get something better near shore that can help with that, but I don't think that data would be useful to look at from an advection perspective.
Thank you.
Wonderful presentation. I had just a general question about would you be able to use atmospheric modeling or predictions to predict zooplankton or marine animal spatial and temporal variability?
You mean in the future?
Yeah. Mm-hmm.
Yeah. Well, actually, Jerome Fischer, who was a collaborator on that and the one running the RUMS Nemusci, has a paper which I think he is finishing to revise that kind of does that, where we're looking at projections from 3 different models in the California current system and using them to force 3 different types of krill models. So my model, Nemusci, and species distribution model to kind of extrapolate krill variability 100 years in the future. So yes, we've done that. The answer is that it's actually interesting how, we didn't do it really to predict krill. It was more in terms of trying to look at what uncertainty you have from different projections versus different formulations. And so that's, hopefully that's going to be out in a couple months and you can look at it. But so yeah, we've done that. I don't know if I would, the different projections do give different answers, so it's a little bit challenging to say definitely this is what's going to happen to krill, but at least we have a better handle on what the uncertainty is, basically.
Great, thank you.
Yeah, so I see how you're getting like these really high variation explanations for your upwelling, but I'm curious what you believe could be some of the variables explaining like the remaining variation that could be happening.
Some of it is likely to be just patchiness of biology, so that when you look at satellite, you're still broad enough scale where you're not going to represent the like variability at the same scale. So that's one thing. I don't know how big it would be relative to just other forcing. Like something like export, just the type of plankton that you have, whether you're looking at a diatom or a small plankton, you might have very different food chains and so very different export after that. All of that was kind of considering one type of phytoplankton, one type of zooplankton. So that's kind of like some future work that I'm hoping to do at some point, is kind of to look at the taxonomy of the phytoplankton and seeing their, like here I'm mostly talking about export, the impact on that variability. For the marine animals, I think there's just so much we don't know about them, like including how they move, like whether that time series is actually representative of a broad region, how much of it is due to just animals moving in and out of that region without having anything to do with upwelling. It's probably a lot too. Interactions between, just food web interactions between who eats who and what happens there. I think there's just, there's just a lot happening. And I'm not a marine biologist, so that I've been working with marine biologists who tell me what might be happening, but it's not something I can easily answer. So.
Thanks.
And do we have any questions on Zoom? No? Okay. I have one last question, which is in the surface water time series, it looked like there was a lot of seasonal variability. If you removed the seasonal cycle, would there be any persistence in those plankton communities that might be explained by the longer term averages in upwelling?
Yeah, we actually looked at that, and I don't remember exactly what the answer was, but we had looked at what I do remember is that we had looked at the seasonal, interannual, and trend in the upwelling time series and looked at how that was explaining variability in the 3 time series, and that was consistently less than the integration, including for the surface. But yes, a lot of that correlation is driven by the seasonal cycle for sure, but you still get some prediction at longer timescales too.
Thank you. All right, well, let's give Monique a hand.